cycle, mitosis, and meiosis. So the cell cycle is basically the cell's life cycle. Cells that are not dividing are in G0, which is considered the resting state of the cell cycle. These are typically cells that, like neurons, that aren't going to divide further. However, certain cells can re-enter the cell life cycle in a phase called G1, which they grow and undergo their normal cellular functions. If the cells need to divide, they'll undergo DNA replication, doubling their DNA content. And then they'll either undergo mitosis or meiosis, depending on some factors I'll talk about later, to split into two, or in the case of meiosis, eventually four daughter cells. So if you have double the DNA content, you don't want that permanently. You're going to have to split and reduce the DNA content of each daughter cell into the same number you started with in the case of mitosis. So the phases are, we're going to ignore G0 for now. G1, basically just growth. S phase, which is when DNA replication occurs. G2 is when the cell is preparing to undergo mitosis in this first case. And then M phase, which is mitosis or meiosis, pardon me. And mitosis can be further divided into some separate stages, which I'll talk about now. So what's the purpose of mitosis? And it's accomplishing the division of one cell into two daughter cells with the same amount of genetic information as the parent cell had. So if you have a diploid cell, the parent cell, so it has one copy of each chromosome, or excuse me, pardon me, two copies of each chromosome, it's going to undergo DNA replication. It's going to end up with 4 copies. It's going to be 4N, so a diploid cell is 2N. It's going to double its DNA and become a 4N cell. And then it's going to head into prophase. This is the beginning of the M stage I was talking about. And the nucleus at this point of the cell where the DNA is starts degrading. And a thing called the kinetochore starts forming. And the kinetochore, you can see it here kind of, or here I have another image, forms from this thing called the centrosome, which is known as a microtubule organizing center. Let's just pretend it's here. It's not really shown in this picture. And then microtubules form from that microtubule organizing center and attach to the chromosomes at a point called the centromere, which is where the two replicated chromatids are attached. So this is a copy of the chromosome, and this is another copy of that chromosome, and they're attached at the centromere. Okay, so back to mitosis. That's prophase. That's when that all starts happening. In metaphase, the chromosomes are all aligned on the equator of the cell in the middle. This is known as the equatorial plate. During anaphase, the kinetochores start separating the chromatids. So this one will go this way, and this one will go this way. Same thing down here. And that pulls apart each of those replicated chromosomes. So you now are reducing your, instead of having double the DNA content, you're returning back to the starting amount of DNA content. And then the last part of mitosis is called telophase. And in telophase, you have this cytoplasm containing both of 4N, so double the amount of DNA content you need. Yes, they're separated via anaphase, but you need this thing called the actin ring. It's a contractile ring, and it basically squeezes this apart right here and divides this one big weird shaped cell into 2 cells. And then these are your 2 daughter cells, which have the same DNA content and composition as the parent cell. So there's no genetic variation here. You're basically just making more of those cells. And this occurs in a lot of biology. If you're just thinking about the human body, you need new skin cells. And so this would be a parent skin cell, like a precursor cell, and it ends up creating more and more daughter cells. These are two separate daughter cells. It's not really doing a good job of this picture showing that they're two different cells. Here's a better one, that are produced from this single cell at the beginning. So that's mitosis. Meiosis is much more complicated. Not to scare you, it just meiosis is different in the fact that you're starting with a diploid cell, and this occurs during sexual reproduction, and you want to create a gamete cell, which it's not diploid, having two of the chromosomes, of two of each chromosome. It's haploid, meaning it only has one of each chromosome. So how this happens is it basically goes through that process of mitosis, similar to that, twice, as you'll see here briefly. Prophase, this thing called prometaphase, which we don't really have to worry about. It's just kind of what happens in between prophase and metaphase. So you have prophase, metaphase, anaphase, and telophase, just like before. You'll notice these are metaphase 1, anaphase 1, telophase 1, because it happens twice. And then down here, you'll see the same process happening again, producing the haploid cells from, let's see, diploid cells. Okay, so how does this work? And why do we care? Meiosis is interesting because it produces some genetic variation. It does this in several ways, and why this is important is when you think about it, you're not identical genetically to your parents. Yes, of course, because you have both your mother and father's DNA combining to form you. That's one way a variation occurs in your genes, but also in meiosis, when creating these gamete cells, you get genetic variation in two other ways. You get crossing over in prophase. So if you have a blue chromosome and a red chromosome, pieces of those chromosomes, when they kind of match up, so they're homologous chromosomes, meaning that they're the same chromosome, but not technically genetically the exact same. So let's say let's call a chromosome 1, just for the sake of calling a chromosome 1. You'll have, in your normal cell, before all this, you'll have a copy of your mother's chromosome 1 and your father's chromosome 1. So you'll have two of these guys right here. Let's say we have a red X and a blue X. The blue X is from your father and the red X is from your mother. And it's the same chromosome, meaning the same genes are held on those. They might be slightly different. And what happens is those two undergo this process called crossing over, in which pieces may splice into the other one and basically trade places, but just parts of it. So you get some hybrid looking like this. And reasons where crossing over occurs are called chiasmata, and that's just a fun factoid to know. So once that occurs, the chromosomes, these little hybrid chromosomes, however, this is pretty much a random process. However this occurred, these chromosomes with the new genetic variation align on the equatorial plate, just like before, in metaphase 1. And then, interestingly, I'm going to get a different picture. Let's see. Meiosis. I didn't really like that one too much. Okay, let's see. How does this one look? Perfect. Okay, so here they are aligned. And the kinetochore forms, attaching the microtubules to the centromeres, the center of the X, if you will. And it decides, in metaphase it's different though. Instead of having each X lined vertically, X, 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 you're paired with, you have an X here and an X here, a chromosome here and a chromosome here, a chromosome here and a chromosome here, as opposed to in mitosis, you just have one vertical line. So here you have basically the blues on this side and the reds on this side. That's just how it's illustrated. But this could be randomized. This one could technically be over here, and this one could technically be over here. These places are variable, and that adds another component of genetic variation, because when the cell splits, certain chromosomes might go this way and certain ones might go this way randomly, kind of like you're shuffling a deck of cards depending on where they originally paired up and lined here. So in addition to crossing over in prophase, you have some more genetic variation being introduced here when the chromosomes actually separate. So this occurs, the actin ring splits the daughter cells. So now you have two daughter cells, and these are diploid cells. So you started diploid, and you're ending diploid. Okay. So this is kind of funky. It's because you want to end haploid, meaning one copy, because that's just how gametes are, because when gametes go to reproduce, they'll combine with the gamete from the other parent to form a new cell, because they have combined. So you'll get a gamete with a haploid number plus a gamete from the other parent with another haploid number combining to create a diploid baby, basically, if you're talking about humans. So what happens here is now the chromosomes align, just like in meiosis. So now they're aligning basically one by one, if you will. They're not paired up like this. They're one by one. So now they split when they get pulled apart by the kinetochore. They get split at the centromere. And one of the copies of the chromosomes goes to each new cell. Now this is kind of a weird how they combine this illustration here. Let's see if I can find another example that might better illustrate this, make it a little simpler. So here you get done with meiosis 1, and then the second division occurs. So you have these two cells. These each undergo meiosis 2, and then you end up with the 4 haploid cells, which end up being gametes. Yeah, so here's a simple example. After DNA replication, you originally had one blue and one pink, I guess you'd say. You go undergo DNA replication to get the double, forming the X, if you will. Crossing over occurs, and then meiosis 1 occurs. Randomly assorting, if there were more, you would see blue going, let's say, to the left and pink going to the right. It could be random, and a shuffling could occur. Genetic variation there. And then here, meiosis 2, you're basically just splitting these diploid cells into haploid cells ready to undergo sexual reproduction. So that's just some basic mitosis and meiosis. I hope that's helpful as a reminder going forward that basically mitosis, you end up with what you start with. Meiosis, you're creating gametes. So you need half the genetic content you would normally want. Therefore, you want haploid cells instead of diploid cells. And in meiosis, genetic variation can occur. And I guess here's an example of some crossing over. I talked about it. This occurs in what phase? In meiosis, right? So in meiosis and prophase, this crossing over occurs, contributing to genetic recombination and variation. Thank you. I hope this is helpful. Until next time.